In this video, we're going to talk about action potentials. An action potential is a rapid change in the voltage across a cell membrane of a neuron, and they're important because they allow cells to communicate with one another. I'm going to first use some masking tape to model both sides of a neuron membrane. So here are the two sides of the membrane. Let's label the inside and the outside. On the other side, I'm going to label a graph. On the y-axis of the graph, I have membrane potential. Membrane potential is measured in millivolts. On the x-axis, I have time. And I'm going to be labeling the three different types of proteins that are on the neural membrane. The first one is a sodium channel. The second one is a potassium channel. And the last one is a sodium-potassium pump. And I'm going to be drawing two more sets of these along each side of the membrane. And now I'm going to add about 30 red pom-poms representing the potassium ions located on the inside of the neuron. And I'm using green pom-poms, about 100 of them, to indicate the numerous sodium ions that are located on the outside of the neuron. So this represents the resting membrane potential, which is referred to as salty banana. I have potassiums on the inside, sodiums on the outside. Since I have way more positive sodium ions on the outside of the membrane compared to the number of potassium ions I have on the inside, the overall membrane potential is a negative number, meaning the inside of the neural membrane is more negative compared to what is on the outside. So the resting membrane potential is negative 70 millivolts.
During an action potential,
an incoming nerve impulse will open up the first set of sodium channels. So sodium ions will rush into the neuron. And same thing will happen from the other side of the neuron. And this influx of all these positive sodium ions will increase the membrane potential, meaning the inside of the neuron is becoming more positive due to all these positive sodium ions. So that number increases all the way up to positive 30. We call this stage of action potential depolarization state. And this change in voltage will open up the nearby potassium channels. So potassium ions will start leaving the neuron. And all these positive potassium ions leaving the neuron will decrease the membrane potential back down to a negative number. So that number now becomes negative 80 millivolts, and this stage is referred to as repolarization state. The membrane potential becomes negative again. And now this action potential will be propagated to the next section of the membrane. So again, sodium ions will enter the cell, and that voltage change causes a depolarization of the membrane. This causes the potassium ions to rush out of the cell, causing the membrane to become repolarized. And the same process repeats again as the action potential gets propagated to the next section. Sodium ions enter, followed by potassium ions leaving. After an action potential, you can see that the original charge and ion distribution is reversed. Potassiums are on the outside and sodiums are on the inside. So we need a way to restore the original resting potential. That's when these sodium-potassium pumps come into play. They're going to restore the original ion distribution. These pumps require ATP hydrolysis. They're going to pump 3 sodium ions out and 2 potassium ions in, and that is moving both ions against the concentration gradient. So it's a form of active transport requiring ATP hydrolysis. And this process will repeat again and again until the original ion distribution is restored. Again, at the resting state, we have all the potassium ions on the inside and sodium ions on the outside. And as you can see, this process requires a lot of ATPs. In fact, about 30% of all the ATPs that our body produces daily is used to power these sodium-potassium pumps. At this time, you may have noticed that there are some potassium ions still on the outside of the cell membrane, and that's okay because the same thing is true about a real neuron at rest. There's so many ions both inside and outside of a neuron, so as long as the overall ion balance is correct, this does not cause a problem. So at this time, the original resting potential is fully restored and the neuron is ready to receive another action potential. But now let's talk about this little dip here on the graph. This is referred to as the refractory period. And this is when your neuron is unable to receive another nerve impulse. This is only temporary, and this happens because, as we saw in our simulation, sodium-potassium pumps, they require some time to restore the original ion distribution. So while the sodium-potassium pumps are still working, the neuron is unable to act upon an incoming action potential.